Welcome to this next video in which we are discussing tuberculosis and anti-tuberculosis drugs. Okay, so we're currently discussing the story of becoming infected with tuberculosis for the first time. And what we've seen so far is that the alveolar macrophages are going to be incapable of controlling the Mycobacterium tuberculosis cells that you have inhaled in on their own. And the reason for this is that when they try to phagocytose them, the engulfing occurs absolutely fine. They get them into a phagosome, but the actual killing inside the phagosome does not occur okay. Instead, Mycobacterium tuberculosis cells have a number of mechanisms for preventing the killing mechanisms from actually occurring. So for instance, they can prevent phagolysosomal fusion, they can prevent the production of reactive oxygen species and reactive nitrogen species, and this means that the Mycobacterium tuberculosis cells can actually live inside the phagosome and not be killed. Worse than that, they can even start dividing inside the phagosome, and once the phagosome becomes big enough and filled up with Mycobacterium tuberculosis cells, it can actually cause the phagocyte, the macrophage here, to actually burst open and release the Mycobacterium tuberculosis cells, and then they will be uptaken by more alveolar macrophages, and the cycle will repeat again and again and again. So the alveolar macrophages have failed to deal with this. And what we now want to discuss is the inflammatory response that is going to occur to try and deal with this. But as we're going to see, it's going to be equally bad at dealing with this. And only once we get the adaptive immune response coming in are we actually going to get the Mycobacterium tuberculosis under control. Okay, so we're now going to discuss then the inflammatory response. So firstly, let me just give you the motivation for the inflammatory response. So the inflammatory response is all about calling for help. It's about calling for help to deal with this pathogen that is present within the tissue. Now, help, troops if you like, to deal with the pathogen is present within the bloodstream. So in the bloodstream you have a number of proteins, for instance the complement proteins, and a number of cells, the white blood cells, also known as the leukocytes, which are all capable of attacking pathogens. The inflammatory response is all about recruiting these troops, recruiting these proteins and these white blood cells from the bloodstream to the tissue where we actually have the pathogen invading. So that's what we're about to watch, what we're about to discuss, this calling for help and this recruitment of troops to try and help control this pathogen that is infecting our tissue. Okay, so it begins then with the calling for help, and this is going to happen with the alveolar macrophages. So actually this will occur long before, for instance, the alveolar macrophages have failed to phagocytose the Mycobacterium tuberculosis correctly. This will occur as soon as the alveolar macrophages actually acknowledge that the Mycobacterium tuberculosis is present. So going back to this piece of paper here, remember the alveolar macrophages recognize the presence of the Mycobacterium tuberculosis using their pattern recognition receptors, which bound to pathogen-associated molecular patterns on the surface of the Mycobacterium tuberculosis cells. And remember, a key example is toll-like receptor 2 on the surface of the alveolar macrophages being activated by lipomannan molecules on the surface of the Mycobacterium tuberculosis cells. So, when the pattern recognition receptors on the surface of the alveolar macrophages were activated, that's when the alveolar macrophages are going to start calling for help. So this happens concurrently with them beginning and failing the phagocytosis process. So I'll just draw an alveolar macrophage here, so the usual great blob of a cell here. And now that it has recognised that there is a problem, it has recognised that there is something very dangerous in the surrounding, what it's now going to start releasing is pro-inflammatory molecules that are effectively the cause for help. So, There are a huge number of different pro-inflammatory mediators. However, if you're going to just learn two, the major, prototypical, most important ones that you need to know about are tumor necrosis factor alpha and interleukin-1. These are the two you cannot get by in immunology without knowing the names of. These are the, the pro-inflammatory mediators to rule them all. They're the most important ones. So let me just write out their full names because they're so important. So TNF-alpha stands for tumor necrosis factor alpha. There are other tumor necrosis factors, but tumor necrosis factor alpha is easily the most important one. And IL-1 stands for interleukin-1. Okay, so these are two really important pro-inflammatory mediators. You might also hear them referred to as pro-inflammatory cytokines. All that is meant by a cytokine is a protein molecule that is released from an immune cell which acts and changes the behavior of other cells. So we can describe tumor necrosis factor alpha and interleukin-1 as pro-inflammatory mediators, if you like, or it's more common actually to hear them described as pro-inflammatory cytokines. So as I'm saying, this word cytokine, all it means is a molecule that's released by an immune system cell, and it has to be a protein molecule. So both tumor necrosis factor alpha and interleukin-1, they're both protein molecules. It's a protein molecule released by a cell of the immune system, which an alveolar macrophage is. It's part of the innate immune system, and which is going to change the behavior of other cells. So these pro-inflammatory cytokines, they're going to change the behavior of cells that make up the micro vasculature, specifically the endothelial cells. So they're going to act on another cell and change the behavior of that cell. So it's just a signaling protein released by a cell of the immune system. That's all that is meant by a cytokine. It's a term that causes a huge amount of confusion as to exactly what it means. So I just wanted to make that absolutely clear. Okay, so the alveolar macrophages, upon recognizing Mycobacterium tuberculosis, then they're going to release these two really important pro-inflammatory cytokines, TNF-alpha and interleukin-1. And these, if you like, are the solid material protein forms of the cry for help. They are a signal saying help, help, help. Call for troops. There is a major problem here. There is something very dangerous here. We need help. Okay, and these molecules, as they say, are going to work on the microvasculature. So their target is the microvasculature, and in the microvasculature they're going to initiate the inflammatory response. So the inflammatory response is a response of the microvasculature. It fundamentally is a change in the microvasculature of the area that has the infection present in it. So in this case, of course, we're working in the lungs, so it's going to be a change in the microvasculature of the lungs. So just to point out where the microvasculature of the lungs would actually be present in my picture of the alveoli here. So in this space in between alveoli here, this is where the microvasculature would be placed. Okay, so in between the alveolar air sacs, that's where the microvasculature blood vessels are going to be placed. So the terminal arterioles, the capillary beds, and the postcapillary venules. So because understanding the microvasculature is absolutely crucial to understanding the inflammatory response, I am just going to include at this point a brief recap of the structure of the microvasculature blood vessels. I think this is really valuable to understanding the inflammatory response, so I'm just going to include this here. So here's my reminder of the microvasculature. So in all tissues of your body, you have microvasculature blood vessels, and the three major microvasculature blood vessels are the terminal arterioles, the capillaries, and the postcapillary venules. And I'm just going to draw a picture of them here. So here is the capillary bed in the middle, and here is my postcapillary venule afterwards. So, to go over this vessel that I'm colouring now in red, this is representing my terminal arteriole. This is an absolutely tiny little blood vessel, a tiny little arteriole, barely bigger than a capillary. And arteriole, I just want to stress this, the word arteriole, it covers a huge scope of different sized blood vessels. Some arterioles are absolutely massive. They're only just smaller than arteries. So the arterioles that come just after an artery, so an artery, a fine or very small artery can then split up into arterioles. Those will be far bigger than this one here. So the word arteriole covers a huge spectrum of different sized blood vessels. I have put the word terminal here to indicate that I am talking about a tiny little arteriole. An arteriole that is the very last arteriole before we then have it splitting into capillaries. So this truly is a tiny little blood vessel, not visible to the naked eye at all. Visible histologically down a microscope. So, then we have the terminal arteriole breaking up into capillaries here, which I'll just colour in green for lack of a better colour. So here I've shown 3 capillaries coming off my terminal arteriole here, and then you can see that the capillaries are reconverging together to make a tiny little venule. And again, the word venule covers a huge spectrum of different sized blood vessels. So what I'm going to do is I'm going to put the term post-capillary in front of the word venule, and this means one of these tiny little venules that is just after capillaries. So the first venule, if you like. So post-capillary venule and terminal arteriole, they're good terms to use to emphasize what size venules and what size arterioles you are actually talking about. Okay, so here is the basic structure of the microvasculature. What I want to do now is actually discuss the structure of each of the walls of these types of blood vessels. So I want to take a cross-section across a terminal arteriole, a cross-section across a capillary, and a cross-section across a post-capillary venule, and show you what each of these blood vessels looks like if we look at it in cross-section and see the different parts of the vessel wall, because that's going to be really important to understanding the inflammatory response. So let's start off with the terminal arteriole. So I'll draw it here. So this can be the lumen of the terminal arteriole, and of course the most inner portion of any blood vessel wall is the endothelium. So I'll just put some endothelial cells here. So here are the endothelial cells lining the lumen of the terminal arteriole. And then of course they will be sat on a basement membrane, which is what I'm going to now put here in red. So the basement membrane is of course holding the endothelial cells in place. They're attached to that. Surrounding the basement membrane, what you then have is a layer of vascular smooth muscle cells in the case of the terminal arteriole. And this is the thing that distinguishes the terminal arteriole from the capillaries and post-capillary venules. As we'll see with the capillaries and post-capillary venules, their wall structure just consists of the endothelium. That's it. With the terminal arteriole, you have an additional layer around the outside, which is made up of vascular smooth muscle cells. So this layer is going to be made up of vascular smooth muscle cells. Now I'll abbreviate vascular smooth muscle cells down to VSMCs. V for vascular, S for smooth, M for muscle, and then C for cells. So if I just draw one of these in here, vascular smooth muscle cells have this spindle shape here, and of course you're going to have whole rings of vascular smooth muscle cells like so. So here I'm drawing lots of vascular smooth muscle cells forming a complete ring like so. And the key message that I want to get across here is that if you can imagine what will happen when all of these vascular smooth muscle cells contract. So imagine them now all contracting. If they were all to contract, then all of their lengths would decrease. Now if all of their lengths decreased, what would happen to the circumference of this ring? Well, the circumference of the ring would go down, and if the circumference of a ring goes down, the diameter of the ring goes down. Showing this with my fingers, if the circumference goes down, the diameter goes down. The lumen of the vessel will become less thick if these vascular smooth muscle cells were to contract. So Arterioles, terminal arterioles, can change their lumen diameter, and therefore they can control the amount of blood flowing through the microvasculature. And that's really important for understanding the inflammatory response, as we'll see. So there's the vessel wall structure of a terminal arteriole. You've got the endothelial layer on the inside, and then surrounding that you've got a vascular smooth muscle cell layer. Now let's have a look at the wall structure of a capillary. So capillaries are even smaller. These, if I were to draw it in scale, would be this sort of size. And it only really takes a single endothelial cell to go around the entire circumference of a capillary. So this endothelial cell still will be on a basement membrane like so. So that is the structure of a capillary. So you literally just have the endothelium with the basement membrane with endothelial cells sitting on top of it, and that's the structure of a capillary. So these are incredibly small little blood vessels. The lumen of a capillary is 1 cell thick. A single red blood cell can fit through here in single file. Okay, as you can see, the lumen of a terminal arteriole is substantially bigger. More than 1 red blood cell could fit through here at once. And the vessel wall is incredibly thin, and this is ideal for gas exchange and other nutrient exchange. Okay, so that's of course where gas exchange and nutrient exchange does occur, and of course in the lungs we'll be getting carbon dioxide leaving the capillaries and going into the alveolar spaces, and hopefully oxygen coming in. Okay, next, the post-capillary venules then. So this will go back to being the same sort of size as a terminal arteriole, but they have a simpler wall structure than terminal arterioles. Let me show this here. So here are the endothelial cells. I'll give them all nuclei like so, and these endothelial cells will again be sat on a basement membrane, which I'll colour in red. But this time, in the case of the post-capillary venules, we're not going to have a layer of vascular smooth muscle cells around them. So the post-capillary venules, the picture is now finished for the post-capillary venules. You're just going to have the endothelial layer, the endothelial cells sitting on the basement membrane, and again we've gone onto a really odd angle. Okay, right, so there's my picture of the cross-sections of these different vessels of the microvasculature: a terminal arteriole, a capillary, and a post-capillary venule. Okay, so we're now ready to discuss the inflammatory response. So you have these microvasculature structures in all the different tissues of your body. You'll have them in lung tissue. These molecules, these calls for help that the alveolar macrophages have released, tumor necrosis factor alpha and interleukin-1, they're going to diffuse away from the alveolar macrophage, and they're going to act on these blood vessels of the microvasculature. And these are the things that they're going to induce. Let's start off with what they're going to do to the terminal arteriole here. So they are going to result in arteriolar vasodilatation. So arteriolar and vasodilatation is the posh old word. The more modern word is just to call it arteriolar vasodilation. So you're going to get arteriolar vasodilation, which means that the vascular smooth muscle cells are going to relax down, and of course if all of them relax, then the circumference of these rings of vascular smooth muscle cells is all going to increase, and therefore the lumen of the blood vessel is going to open up. Now interleukin-1 and tumor necrosis factor alpha, they don't act directly on the vascular smooth muscle cells. So they don't act directly on receptors on the vascular smooth muscle cells to cause relaxation. Instead, they actually act on the endothelial cells, which then send molecules to the vascular smooth muscle cells to tell them to relax. But we won't fuss over the details. Overall, when tumor necrosis factor alpha and interleukin-1 go up in the area, it's going to cause arteriolar vasodilatation. The arteriole lumen, the lumen of these terminal arterioles, is going to increase. Now, what is the point of that? Well, the point of that is it's going to lead to increased blood flow. So the blood flow to the area where the Mycobacterium tuberculosis is causing a problem is going to go up. Now, why exactly would you want to increase blood flow to the area where the Mycobacterium tuberculosis is? Well, remember what the point of an inflammatory response is. We're trying to recruit troops from the blood to the area to help deal with the infection. Okay? If you've got more troops flowing through an area at any one time, so if blood flow has increased, you have more troops that you could potentially recruit from that blood to the affected area. So the idea is that if we increase blood flow, the recruitment of proteins from the bloodstream and white blood cells from the bloodstream is going to be increased. Okay, so that's the reason we want terminal arterioles to vasodilatate. Next, we're also going to get what's known as endothelial cell contraction. So, interleukin-1 and tumor necrosis factor alpha, they cause endothelial cell contraction. Now, what does this mean? This means that the endothelial cells are going to open up gaps in between them. And by the way, endothelial contraction is going to occur in the capillaries and the post-capillary venules. So I'll just fill in this label here. So this is a post-capillary venule. So arteriolar vasodilatation was obviously a change that occurred in the terminal arterioles. Endothelial contraction is going to be a change that occurs in the capillaries and the post-capillary venules. So let me just draw you a little picture of endothelial cell contraction. So, if I draw a picture of a bit of endothelium of either a capillary or a post-capillary venule, it looks like this. Here is an endothelial cell, here's another endothelial cell. I'll just give them nuclei, and they're both sitting on the basement membrane like so. Now, initially, the endothelial cells, they don't have very big gaps in between them. Indeed, there are proteins in the membranes of each of the endothelial cells that are bound to each other and which are keeping these two membranes tightly opposed to one another to close up this potential gap between neighbouring endothelial cells. So initially, in the capillaries and the post-capillary venules, the gaps in between the endothelial cells are tightly closed. What's going to happen in endothelial cell contraction initiated by interleukin-1 and tumor necrosis factor alpha at the site of infection is the endothelial cells are going to pull apart. They're going to break these junctions in between their opposing membranes, and they're going to open up gaps in between them, each other. So here's one endothelial cell, and here now is its neighbour, and you can see that they have pulled back to open up a gap in between each other. Okay, so the endothelial cells contract backwards to open up gaps in between themselves, and therefore you're going to allow fluid from the bloodstream to leave the bloodstream and go into the tissue fluid, go into the peripheral tissue. So what is the purpose of endothelial cell contraction in the capillaries and post-capillary venules at this affected site? Well, it forms what is known as an inflammatory exudate. So this is responsible for the formation of an inflammatory exudate at the site of infection. And an inflammatory exudate is fluid accumulation at the site of infection. And this fluid contains a lot of the proteins from within the bloodstream. So these gaps allow proteins from within the bloodstream to leave the bloodstream and go into the tissue fluid of the affected tissue. So they're going to go into the tissue fluid of the lungs in this case. And this will bring in complement proteins and other proteins within the bloodstream that are capable of attacking pathogens into the affected area. So the idea behind endothelial cell contraction is that you form an exudate which contains proteins from within the bloodstream capable of attacking pathogens. So it's another part of bringing in troops. Well, this is part of bringing in troops as part of the inflammatory response. Now, endothelial cell contraction alone is not actually enough to get white blood cells recruited from the bloodstream into the peripheral tissue fluid, because these gaps aren't actually big enough for the white blood cells to get through, at least not with a lot of help. So what also interleukin-1 and tumor necrosis factor alpha are going to do to the endothelial cells of the capillaries and post-capillary venules is they're going to get them to start participating in the process of leukocyte recruitment. And remember, leukocyte, either spelt with a K or a C, it doesn't matter, is another word for white blood cell. Okay, so we're going to start recruiting white blood cells to the affected area. So this is another part of the acute inflammatory response. Okay, so in order to participate in leukocyte recruitment, the endothelial cells of the capillaries and the postcapillary venules, they have to start putting all sorts of special proteins on their cell membrane, which can interact with proteins on the surface of the white blood cell, and which will help the white blood cell to move through the gaps between the endothelial cells. So let me just show this here. So what is actually going to happen when we recruit a white blood cell is something along the lines of this. So here are two endothelial cells still sitting on their basement membrane, and of course we can see that they've contracted, so they've opened up this gap in between them. And now let me draw the process of leukocyte recruitment. So let's have a leukocyte here that's being recruited, and we might say that this is a neutrophil that's being recruited, so it would have a very strange shaped nucleus. So neutrophils have this classic trilobed nucleus. They don't have three nuclei, but their nucleus is a really strange shape. It has these three lobes connected by these little interconnections between the lobules, these little tubes if you like. So here's a neutrophil, and in order for it to be recruited from the bloodstream, it has to squeeze through this gap like so. And this process of squeezing through the interendothelial cell gap is known as diapedesis. And in order to get them to do this, they have to be interacting with loads of proteins on the surface of the neighbouring endothelial cells. So the neighbouring endothelial cells have to put these proteins on their surface, and they will do so in response to interleukin-1 and tumour necrosis factor alpha. Okay, so the endothelial cells are there in green, the neutrophil is here in orange. Okay, so you start getting leukocyte recruitment also as part of the inflammatory response. Now, which sort of leukocytes are going to be recruited majorly as part of the inflammatory response? Well, one of them is the neutrophil. The other type is the monocyte. Okay, so let me just explain what monocytes are. So monocytes, in contrast to neutrophils, they have one nucleus, not one nucleus, a nucleus with a single lobe. They do have one nucleus, but remember the neutrophils have one nucleus as well. The difference between monocytes and neutrophils is that they have a nucleus with a single lobe, like so. So monocytes are a type of white blood cell that circulate within the bloodstream, and they are the precursors to macrophages. So you do not have macrophages circulating within the bloodstream. Instead you have monocytes. These can be recruited from the bloodstream into tissue fluid at peripheral sites, and then once they've been recruited they will then differentiate into macrophages, and they'll become these great blobs of cells, like so. So monocytes can differentiate into macrophages. So the two major types of white blood cell that you're going to recruit from the bloodstream as part of the inflammatory response are neutrophils and macrophages. And neutrophils and macrophages, they are both phagocytes. So their major way of clearing pathogens is by phagocytosis, by ingesting or engulfing the pathogen and then attempting to digest it down. Now, the question then is this inflammatory response going to help us with clearing the Mycobacterium tuberculosis? Well, of course the answer is it's going to do something, but then I suppose the better question is it going to clear the infection? And the answer is no. So overall, looking at what we've achieved here, we've brought in an inflammatory exudate, and as well as that, we've brought in a huge amount of neutrophils and a huge amount of macrophages. Okay, so in fact, I might just draw a little bit of the alveolus that has actually undergone this inflammatory response. So in the lung tissue, and I'll just draw a little bit of the alveolus back again here. So here is our alveolus back again. Okay, we'll now have again our type 2 pneumocytes here, and they'll be starting to be disturbed by all the cells that we're bringing in, because the cells will have to get into the alveolar air spaces. They can't just remain underneath the alveolar air spaces in the tissue fluid, they've got to get into the alveolar air spaces. So the pneumocytes will be making way for the cells coming through here. So here are a few of the pneumocytes, like so. These are the type 1 pneumocytes. Here's another type 2 pneumocyte here. Okay, so we'll now be bringing in a huge number of macrophages and neutrophils, and I'm just going to draw these in here, accumulating in the alveolar spaces. So these can be neutrophils. We'll also be bringing in a lot of inflammatory exudates, so you'll be getting fluid filling the alveolar spaces as well. Okay, so these are all neutrophils, and I'll colour those in orange here, and then I'll put in more macrophages as well. So here then we'll have a new macrophage that we've brought in here, another new macrophage that we've brought in here. So we're bringing in lots of inflammatory cells, lots of these phagocytes, neutrophils and macrophages, and of course inflammatory exudate is going to accumulate in the portion of the lung where we have actually had the inflammatory response take place and the Mycobacterium tuberculosis infecting. Now, what's actually going to be happening? Is this going to be doing any good? Well, of course, all that's going to be continually happening is we're going to be going round and round in this cycle where phagocytes are going to take up the Mycobacterium tuberculosis into phagosomes, and then they're going to fail to kill them. The Mycobacterium tuberculosis will then divide inside the phagosomes, burst the phagocyte open, and then move on to new phagocytes. Okay, occasionally a phagocyte will get the better of the Mycobacterium tuberculosis and actually achieve killing of that Mycobacterium tuberculosis. However, often the Mycobacterium tuberculosis bacterial cell will get the better of the phagocyte, so gradually it's getting out of control, so the inflammatory response is not going to help. I should also stress that neutrophils, they are not better phagocytes than macrophages. Macrophages, if anything, are the stronger phagocytes than neutrophils. Neutrophils are worse at phagocytosis than macrophages. So if macrophages can't handle the Mycobacterium tuberculosis, the neutrophils aren't really going to be able to handle the Mycobacterium tuberculosis. I mean, they'll do their best, they will have a go, but they'll be beaten by the Mycobacterium tuberculosis just as the alveolar macrophages were. So overall, at the end of this video, we've got to the stage of the story where we've had this inflammatory response occur in our lung tissue. We've brought in an inflammatory exudate, we've brought in neutrophils and macrophages, and still we can't really control the Mycobacterium tuberculosis. Occasionally our phagocytes are getting the better of the Mycobacterium tuberculosis and actually managing to kill it, but often the Mycobacterium tuberculosis will live inside the phagosome, breed inside the phagosome, and therefore overall the population is going up. So the rate at which we are getting the division of Mycobacterium tuberculosis is beating the rate at which we are killing Mycobacterium tuberculosis, and overall there is not a balance. So I'm just going to write this down. So at the moment, the rate of production of new Mycobacterium tuberculosis cells is exceeding the rate at which we are killing Mycobacterium tuberculosis cells. So it's not stable at all at the moment. We do not have a dynamic equilibrium. Instead, the population of Mycobacterium tuberculosis is going up, 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 which is a big, big problem. So what we're now going to see in the next video is we are going to launch an adaptive immune response, and the adaptive immune response is then finally what will bring this under control. But what you're going to have to understand is even the adaptive immune response cannot make this bigger than this in most cases. Usually what the adaptive immune response achieves is it makes this equal to this and establishes an equilibrium that goes on and on and on for years, and that's what latent tuberculosis infection is. But more of that later on. So in the next video we'll start the adaptive immune response.